I'm very grateful for the warm welcome, and it's a pleasure to be speaking to you all this afternoon. What I'd like to do is to share with you some of the work in my lab over the last 10 years or so concerning cancer suppressor mechanisms that guard the human genome. So let me start by trying to tell you why I got interested in a particular problem and what it is that I've been trying to understand over the last decade or so. I think if you look at it from the perspective of you and me, that is human beings, you would say that cancer is a very common disease. Why is that? Because about 1 in 3 of us is going to get cancer, and about 1 in 4 of us is unfortunately going to die from cancer. So from a human perspective, this is a common disease. But I've been interested from a different perspective. And that is from the perspective of cells that make up our bodies. So think about it in this way. There are hundreds and hundreds of millions of cells in our body, any one of which can transform and become a malignant cell. So why is cancer so rare? Why is it that only 1/3 of us get cancer over a lifetime of 60 or 70 years? And clinicians will tell you another fact, which is that only about 1 in 10 or 1 in 20 pre-malignant lesions, now I don't mean cells, I mean things you can see, so many cells, but even those lesions, only about 1 in 10 or 1 in 20 will become tumors. Why is that? I think what these observations are telling us is that our cells and our bodies must have very complex systems whose purpose it is to prevent the transformation of cells into cancer cells. And I've been interested in understanding these mechanisms because I genuinely believe that understanding these will give us clues to how to treat cancer and perhaps even more importantly, prevent it from occurring at all. So let me give you a sense of where this work started almost exactly now 20 years ago in 1995, 1996. It's been known for a long time that if you take common types of epithelial cancer, cancers of the lung, the breast, ovary, the prostate, in other words, all of the common forms of cancer, about 10% of them run in families, suggesting the genetic component. But about 90% of them are sporadic, you can't predict. In 1995-96, there was an exciting moment in the field when the first 2 germline cancer susceptibility genes predisposing to a common form of epithelial cancer were cloned. And these are the genes called BRCA1 and BRCA2, or breast cancer 1 and breast cancer 2, as I said, between 1994 and 1996. Now what we know is that these genes account for about 50% of the familial cases. But what was really interesting for me at that time is these are high penetrance genes. What do I mean by that? I mean that if you have a mutation in one of these genes inherited in your germline DNA, you have roughly a 70% chance of developing some form of cancer by the age of about 55 or 60 years. So you see immediately why I was excited. I want to understand why we don't get cancer. And here is a naturally occurring mutation in people who do get cancer much more frequently and much more early than the rest of us. So we felt immediately that if we were able to understand how this gene works, then we would understand or have a clue to the problem that I'm interested in. So to begin with, let me tell you about the gene product that we focused on, which is the BRCA2 tumor suppressor gene. You'll see that it's a very long polypeptide, over 3,500 amino acids. Its molecular mass is apparently over 400 kilodaltons in its native state. But remarkably, for a protein so large, there is nothing in the protein sequence that tells you about its function. And this was both exciting but also discouraging. How do you work with a protein so large and try to understand what it is doing if there are no clues from the sequence? So we turned in the first instance to look again at the patients. And patients, if they inherit one mutant allele, one of the two copies of BRCA2 is mutated, they are predisposed not just to breast and ovarian cancers, but also pancreatic, prostate, male breast, lots of other cancers. Quite peculiarly, if you inherit one mutant allele, you're predisposed to cancer. But if you look in the cancer cells from these individuals, the second copy is thought to be lost, which suggests that it is inactivation of the function of this gene in its entirety that promotes cancer in these cells. Are you with me so far? So that's what we had to do, which is to try and define an experiment which would tell us something about function from this perspective. And so we turned to a tool that has been widely used now, which is transgenic mouse models, in which you can engineer the genes within the mouse germline and recreate models of disease. And what we did, working with colleagues in Cambridge, Martin Evans and Bruce Ponder, was to create mice that were lacking both copies of BRCA2, just as the cancers in these patients. And this is what we found in work that was actually done by one of my postdoctoral colleagues, who is now a distinguished investigator in his own right, Ketan Patel. So what we found is that when you look at cells from these mice lacking BRCA2, their chromosomes undergo very grotesque abnormalities of shape and number, which you can see here in this panel. Normal mouse chromosomes should look like that. But can you see at the arrows how many abnormal chromosomes there are? And what's also interesting is that many of these chromosome abnormalities have a particular structural significance. For example, can you see that break that affects just one of the two sister chromatids? That type of lesion means whatever the problem is, it is probably happening during DNA replication, when the cells are dividing. And these problems in the cells appear before there are any tumors in the mice. And so it struck us that this might be the heart of the cause of cancer in mice lacking BRCA2. I suspect that 99% of you in this audience will know exactly what chromosomes are, so I hope that you will forgive me if for the 1% I just explain what I mean. Chromosomes are essentially, in my view, the physical and functional containers of the human genome. You have the DNA strands, which encode different codons or letters, so to speak. And these are packaged, if you will, into a book, which is what the chromosomes are. They package these information containing strands of DNA. And the point is that this information is not static. It's not in a museum when it's in a cell. It's being used, accessed all the time. It has to be read, it has to be copied when cells divide. So in this sense, the 46 human chromosomes have to be the functional containers of this information and allow access, but at the same time provide protection when it's needed. And indeed, chromosomes are vulnerable to damage by many sorts of exogenous factors as well as endogenous factors. When cells divide, the chromosomes have to be unpacked, the DNA has to be copied, then repackaged. There are external energies, like for example, ultraviolet radiation, X-rays, and so on, which can break the DNA double strand or cause nucleotide abnormalities which damage the DNA. And in all this, the chromosomes and the material which packages the DNA serve as the guardians, if you will, of the human genome. But what we had found in essence was that BRCA2 was a factor that was a guardian of the chromosomes, prompting us to ask this question: who guards the guardians? How are chromosomes protected in order to maintain the integrity of DNA information in cells as they go about their business? And since I started working on this problem, that's the only thing I've worked on in my lab, because as I hope I can share with you, this is a fascinating problem with many interesting answers, highly relevant to human diseases like cancer. So, what does BRCA2 do to guard genetic information within chromosomes? Well, we know that breaks in DNA, double-strand DNA breaks, are amongst the most toxic and mutagenic of lesions. In mammalian cells, they are normally repaired by two mechanisms. One, which is called non-homologous end joining, is a simple sticking together process that is very error-prone because you can change nucleotides at the break junction. A second process, which is called homologous recombination, is intrinsically error-free. It works by exchanging genetic information between the damaged chromatid or strand and an undamaged copy, which is usually on the sister chromatid. So now perhaps you can see what process is likely to be affected in BRCA2 deficient cells, because I showed you breaks that affected just one sister chromatid. And indeed, that is what we and several other labs were able to show, that BRCA2 is essential for the process of error-free repair by homologous recombination, but not for the error-prone process of non-homologous end joining. So that prompted us in the lab to begin to ask, how does BRCA2 work? How does it promote this error-free repair of genomic information? And this is a bit of background that I have to give you. So if you look here, what you see is the break. And if you look here, you see the end product of the repair, which is a gene conversion event which exchanges DNA to repair that break. We still know very little of the steps that go from there to there. But we do know one thing. We know that the first step in this repair reaction is to chew back one of those ends and leave a long overhanging single strand of DNA. And that overhanging single strand then becomes coated with this protein, RAD51, which is a small, highly conserved protein which forms very ordered helical filaments on DNA, so-called nucleoprotein filaments, DNA protein filaments. And we know that this filament is essential for the strand exchange reactions that lead to repair by homologous repair. So Veronica Yu, one of the postdocs in my lab at the time, made the very important observation that in cells lacking BRCA2, these RAD51 multimers fail to assemble at the sites of DNA breakage, which suggested to us that BRCA2 must be controlling RAD51 at the site of repair. So that led us to work with Luca Pellegrini and Tom Blundell in the Department of Biochemistry in Cambridge to solve a crystal structure for a complex between RAD51, which is shown for you here in magenta and blue, and the piece of BRCA2 shown in green that binds to it. Mention, and some of you may have missed, the fact that within the middle of BRCA2 are 8 short repeated protein stretches. They are called the BRC repeats. There are 8 of them, and they're highly conserved in all mammalian cells, both in spacing and in sequence across all mammalian species, which suggests that they're very important because they've been conserved for several millions of years of evolution. It's that part of BRCA2 shown for you here which binds to RAD51. And you can see from this crystal structure that in the middle of RAD51 is this flat beta sheet. And what BRCA2 interaction does is to extend that sheet by an extra 2 strands. And in fact, most of the important contacts between the 2 proteins are made against these anti-parallel strands, which I'm going to enlarge for you now to take you to the next part of my talk. So what we found essentially was that 2 hydrophobic residues, a phenylalanine and alanine, thank you, were critical in making these contacts. And the moment we understood this, we could begin to look in other proteins for the occurrence of this interacting motif. And to our surprise, we found this phenylalanine-alanine motif not just in BRCA2 across all mammalian species, we found it in RAD51. Isn't that beautiful? So you see, that sequence actually occurs within the RAD51 itself. And it goes back in evolution all the way to the 3rd kingdom of life, to archaea. So hundreds of millions of years of evolution. Again suggesting that this is somehow very important. So these results prompted us to propose the following mechanism for the action of BRCA2 in controlling RAD51. I remind you that I said that RAD51, in order to work, has to make multimers on DNA. So we posited that the protomer-protomer interface that enabled multimerization was this FA motif. And that a protein coming later in evolution, BRCA2, had mimicked this motif. And by mimicking, could control the assembly of the RAD51 filament. Does that hypothesis make sense to you? So, needless to say, after we published this work in 2002, it's taken us nearly 7 years of effort to sort out the real story. And of course, it's not exactly how we pictured it. And the reason, I hope, is quite obvious. So here I'm depicting a model in which BRCA2 BRC repeats are inhibiting the formation of this active RAD51 filament. But I told you a few minutes ago that BRCA2 is necessary for RAD51 to work. So how can we reconcile these 2 observations? And that's what's taken us nearly 7 years. So the breakthrough we had in my lab was through protein biochemistry. And again, perhaps I can emphasize that in trying to tackle problems of this kind, one needs to be able and willing to move out of your comfort zone and do experiments and learn methods that are not necessarily the ones that the entire lab works on. And here, Mahmood Shivji, a postdoc in my lab, and Owen Davies, a PhD student joined with biochemistry, made the very important step of expressing and purifying a large segment of BRCA2, which contains all of the 8 BRC repeats. This is a protein fragment. You might call it a fragment, but it's 1,028 amino acids long, which is more than many big proteins. And indeed, they were able to purify this to homogeneity, as you can see, using biochemical techniques. And that enabled us then to begin to ask the question, how do the BRC repeats actually work in regulating RAD51? Now, because your Vice Chancellor is a very distinguished physicist, I couldn't resist putting in one data slide, which again reflects that we have, in a very primitive way in biology, had to use physical techniques, and they have greatly enriched our ability to study the biochemistry of chemical reactions happening inside cells. So the method we're using here is two-color coincidence detection. And this was a collaboration with David Klenneman's lab in chemistry. So what we're doing here is essentially to label two of the interacting species. We have single-stranded DNA, we have double-stranded DNA, we have RAD51, and what we have done is to label the DNA and the RAD51 with spectrally distinct fluorophores. We can now in a test tube allow reactions to proceed, but at a concentration wherein if we illuminate with intersecting confocal beams a very, very tiny volume, femtoliters, then we can count the fluorescence pulses as molecules and complexes of molecules diffuse through this space. These fluorescence bursts in the two spectra will indicate the presence of the molecule, and their amplitude gives us some indication of number. But essentially, because these solutions are sufficiently dilute, if there is intermolecular binding, then there is coincidence, as you can see there, which means the two molecules are together and therefore physically associating. And this is a true single molecule technique for biological chemical reactions. And what I hope you can see is it's extraordinarily informative. In physiological ionic conditions, if you take single-stranded DNA and put in RAD51, to our surprise, RAD51 hardly binds. But if you add in the BRC repeats of BRCA2, then you get a clear increase in binding as well as in the average number of molecules of RAD51 bound to fragments of single-stranded DNA. So in other words, we could tell from these analyses that BRCA2 was contrary to our expectation, promoting, not inhibiting, the binding of RAD51 with single-stranded DNA. So Mahmood and Shreyas went on to repeat these experiments with double-stranded DNA, and they found exactly the opposite. They found that BRCA2 inhibits RAD51 binding to double-stranded DNA. And it's these two observations that gave rise to the model that we were able to propose in 2009, and this includes work that was in collaboration with Steve Kolchowski's lab at the University of California in Davis. But the model is as follows. So look on this side. Here we don't have BRCA2. We just have RAD51, single-stranded DNA, and double-stranded DNA. What happens? Well, what happens is that the RAD51 binds to the wrong substrate. It's very avid in its binding on double-stranded DNA, but of course that is the second step, not the first step of this reaction. And so this reaction is sterile or at best inefficient. But the presence of BRCA2 with these opposing effects, inhibition on double-strand means, and promotion of single-strand binding, means that you get the single-strand nucleoprotein filament first, and it can then engage the double-strand to give rise to the joint molecules that lead to efficient strand exchange. So as I hope I've convinced you, this work tells us that our original thoughts were very primitive compared to the complexity of the way that cells work. And even here, this work is in vitro using a completely reconstituted reaction, purified biochemical components in vitro. So we're still now using spectroscopic techniques, but now in living cells, to be able to understand the dynamics of this reaction in cells undergoing DNA damage. And that's ongoing work in our laboratory. So in summary, BRCA2 is an important guardian of chromosomes through its role in DNA repair by homologous recombination. And I hope with these three experiments over a long period that I've shared with you, you understand the work we did and how we arrived at experimental data which tells us what we think BRCA2 is doing. Let me also emphasize how multidisciplinary work had to be. We could not have done this just being biologists. And that's been a very exciting learning curve for me and for the people in my lab, which has really led over the years to the type of laboratory that we've constructed now in Cambridge. So I was talking about these results in a hospital audience, and one of the clinicians said, "Very nice, but what does it mean for me? I treat patients who have BRCA2 deficient tumors." And this prompted Ferdinando Skoulidis, a clinical PhD fellow in my lab, to embark on experiments to set up animal models that faithfully recapitulate what is going on in BRCA2 mutation carriers to further our laboratory understanding towards in vivo systems. So we didn't choose to work on breast cancer, we chose to work on pancreatic cancer for two reasons. The first is, as you can see, people who inherit BRCA2 mutations have a high excess of pancreatic cancer incidence. It's a very deadly tumor. Only about 10 to 20% of people who present in the clinic have surgically resectable pancreatic cancer. The other 80% are not going to survive for more than a few years. We don't know how to treat it, so it's important to understand how it occurs. But the second is, we know that within these tumors, besides the BRCA2 mutations, you get activation of oncogenes like KRAS occurring consistently across these malignancies, which gives us clues as to how to model this in transgenic mice. So what Ferdinando's did was this somewhat heroic experiment in which he constructed a transgenic mouse strain which had, you can see here, how many different genetically engineered alleles all in the same mouse. So first, below the dashed line, is a truncating mutant in BRCA2, which is the germline mutation in all cells which people inherit when they inherit BRCA2 mutations. Everything here is only in the pancreas. It's engineered to only occur in the pancreas using what's called the LOXP-CRE recombination reaction. We can activate KRAS, we can inactivate P53, and we can inactivate the second copy of BRCA2 only in the pancreas using this PDX1 CRE transgene, which allows recombination of these alleles only in the pancreas. So by this combination of tissue-specific and general alleles, we were able to faithfully mimic what happens in patients with pancreatic cancer. Well, the first thing to tell you is this is a very, very good model for human pancreatic cancer. The tissues look exactly as you would see in human pancreatic cancer. The progression from early lesions to late lesions is exactly the same. The clinical behavior of these tumors with local invasion and metastases into organs like the liver and the lung is just the same as you see in the human disease. So it's a very useful model for us to begin to test treatments and other aspects. Second, we had a very unexpected observation. Now I'm going to spend a minute on this, but rather than talk you through both of these panels, I'm going to concentrate on this panel, but please take my word that both have the same information, but in slightly different allelic strains, which I won't trouble you with. So here you have death of mice over time occurring in the blue line without any mutation in BRCA2. So you see, because of the KRAS oncogene, these mice develop pancreatic cancer and they die in about 250 to 300 days. Now, if you put in one mutant BRCA2 allele, which is the inherited BRCA2 allele, you get acceleration of tumorigenesis. If you put in two mutant BRCA2 alleles, you get much more acceleration of the tumorigenesis, confirming to us that BRCA2 really is preventing or slowing down tumors in these tissues. But what was really interesting for us is this, where you have only one allele of BRCA2 missing. What happens there? Now, what we thought was that BRCA2 behaves like a classical tumor suppressor gene. Al Knudsen in the 1970s, working on genes like retinoblastoma or RB, made the hypothesis that many tumor suppressor genes need two hits. You inherit one germline hit, and that's throughout your body. The second hit occurs only in the tumor tissues, but you need both hits to get tumors. So we thought that these mice, if we looked inside the tumors, would have developed the second hit. Not so. We examined tens of tumors from these mice, and we have yet to find tumors that have lost the second allele. In all of them, and I show you here southern blots at the level of DNA, qPCR reactions at the level of RNA, and crucially, these are Western blots to indicate that full-length protein is present. In fact, we've also sequenced the other alleles, and we know that there are no subtle mutations in these alleles. These are wild-type BRCA2 alleles, yet the mice are getting pancreatic cancer. So this immediately tells us that BRCA2 is not working in the classical mode like a two-hit tumor suppressor. It must be working in a different way. And so the model that we have come to was that in this system of KRAS-activated pancreatic cancer, that the loss of the second BRCA2 allele is unnecessary for the development of tumors. You might ask, as my physician friend did, so what? These are mice, we're people. So we collaborated with Jan and Jorun at the Icelandic Cancer Registry in Reykjavik, because in Iceland, a high percentage of the population have a founder mutation in BRCA2. And so we could find cases in their archive in families carrying this mutation who had developed pancreatic cancer. And what I'm showing you here is a small number of cases. We have since enlarged this, but at the moment we find that a significant fraction, somewhere between 50 and 75% of these patients, do not lose the second allele, just as in mice. So this is turning upside down our consideration of how BRCA2 works to protect chromosomes, and we're still in the throes of investigating the mechanisms behind the effects of heterozygosity. But what I'd really quickly like to take you through is that it has two important implications. And the first is, it enabled us, using this model, to propose a possible answer to this question. I've told you that BRCA2 is involved in protecting chromosomes. I've told you that's involved in DNA repair. But these are functions that are present or necessary for the viability of any cell in the body. So why do patients carrying these mutations get pancreatic, breast, ovarian, and prostate cancers? Why the tissue specificity? So we put together several aspects of our cellular work to posit the following model, and then tested it using the mouse model that I just described to you. So what we argued to ourselves was, people inherit one mutant BRCA2 allele, one wild-type BRCA2 allele. We assume that as the second allele can be lost in any tissue through stochastic genetic mutations that occur in all cells. We were able to show in vitro, in work that I've not had time to share with you, that loss of that second BRCA2 allele in fact leads to cell death. And you can understand why. I showed you those shattered, broken chromosomes in BRCA2 deficient cells. They're going to die. So why do these mice get tumors? Our hypothesis is that only in certain tissues can these cells survive long enough to accumulate second mutations. And those second mutations, which have been found in patients' tumors, include mutations in genes like p53, whose job it is to detect broken chromosomes and eliminate those cells. But cells without p53 are much less able to respond and therefore can continue to proliferate, and we and others have been able to show that BRCA2 deficient cells in vitro, if you put in a p53 mutation, can continue to divide despite their broken chromosomes. But if only certain tissues allow this prolonged survival, then that may explain why only certain tissues can give rise to this type of tissue-specific expression. To partially, I emphasize partially, test this idea, we turned again to our mice, and I draw your attention now to this panel. So what we did was here, we activated KRAS, we eliminated one allele of BRCA2, and then we asked, what happens if we eliminate the second allele of BRCA2? Do we get tumors? And in fact, what you can see is if you eliminate the second allele of BRCA2, you don't get tumors, you get no pancreas. The mice die because all the pancreatic cells undergo cell death because of the broken chromosomes. But if you now, at the same time, put in a mutant p53 allele, then, and only then, you get these tumors, which partly confirms that that second mutation is necessary for the outgrowth of tumors in an in vivo setting. And now we are testing the idea that only the target tissues allow the survival of these cells lacking both alleles of BRCA2 long enough to acquire these types of mutations. There's a second implication of the model, and that directly relates to what my physician friend talked about. How can we use this type of information to better treat tumors arising in BRCA2 mutation carriers? So to tell you about this, I have to introduce you to another concept, which is again a very fundamental issue. I think you all know that cells divide by replicating their DNA. You also know about DNA polymerases, the leading and the lagging strand polymerases. And you know that these are very rapidly acting enzymes which can copy long stretches of DNA very quickly and with very high fidelity. But what perhaps many of you may not have appreciated is these enzymes encounter very frequently, even in normal cells, roadblocks to their progress. These roadblocks could be nucleotide lesions, for example, UV-induced nucleotide lesions. They could be proteins that are tightly bound to the template and cannot be displaced by replicative helicases. They could be interstrand crosslinks between the strands of DNA, which some of the chemicals that we encounter in the environment induce. But the point is that every organism exposed even to UV, like E. coli to us, will have to have systems to restart stalled replication. And what we were able to show is that BRCA2 has an essential role in restarting stalled replication. So in the absence of BRCA2, our work showed you don't get restart, but instead what you get is DNA breaks that lead to cell death. And this was work that one of my postdoctoral fellows, Misha Lomonosov, did about 10 years ago. But we were able to quickly take advantage of that idea and say, supposing we treated these cells with chemotherapeutic agents that block DNA replication. For example, mitomycin C, carboplatin, these are compounds already in use in the clinic. Are they more sensitive? The answer is yes. These cells are 50-fold more sensitive to these cross-linking agents than our normal BRCA2 proficient cells. And it's been immensely satisfying to read in the last 6 months the reports of the first big clinical trial of ovarian cancer in BRCA1 patients treated with carboplatin, where clinical responses, partial clinical responses, these are very serious tumors, are observed in over 83% of these women. Even better, some really terrific work from Thomas Halliday's lab in Oxford and Alan Ashworth's lab in London found another way to block replication in BRCA deficient cells. They developed inhibitors of an enzyme called PARP1, poly-ADP ribose polymerase 1, and what this inhibitor does is to block a type of DNA repair that leaves little gaps in the DNA that effectively stalled replication. And BRCA deficient cells were hundreds of times more sensitive to these inhibitors than were normal cells. But the problem here, which I hope those of you who have been reading the title of the slide, which I've deliberately left to the end, is as follows. You can only see the sensitivity in cells which are homozygous, lacking both copies of BRCA2. You don't see these effects in heterozygous cells. But I told you, in our pancreatic model system, in patients with inherited pancreatic cancer, a significant fraction of these tumors have not lost the second allele. Obviously, we thought they must be resistant to PARP inhibitors. And indeed, when we checked the tumors from the heterozygous mice, which are shown for you here, are indeed far more resistant to this drug, which is the AstraZeneca olaparib as PARP inhibitor, which is now in clinical trials, compared to the homozygous mutant mice. So there is resistance. And unfortunately, this does affect the way new approaches for therapy of these tumors are evolving. So we know already from the phase 1 trials of compounds like olaparib that they're well tolerated, they're efficacious in BRCA mutation carriers, and there's a high objective response rate of over half of the patients responding. But that shouldn't mask the fact that nearly half of these patients are not responding. All of them are BRCA mutation carriers. All of them should, in theory, be responding. And that's prompted us to ask the question whether, in fact, if some of those tumors have not lost the second allele, is that what is causing primary resistance to this new drug? And could it be a significant problem in clinical trials? In other words, you should not be selecting, we think, the patients just based on their family history, just based on analyzing their blood for the presence of the heterozygous BRCA mutation. You may have to look in the tumor to check that it has lost the second allele on the diagnostic biopsy. We worry that if you don't do this, then you may get poor results in clinical trials. So this is a trial done by my colleague Stan Kay at the Institute of Cancer Research in London, which was a comparison between a PARP inhibitor, olaparib, and a very simple therapy, which is an anthracycline, a DNA damaging agent, which is widely used and is very cheap compared to new targeted inhibitors. And at least in this study, which was not stratified for homozygous loss, this was just all BRCA mutant patients, unfortunately, they didn't find a statistically significant difference in the response rates between PARP inhibitor and the simple drug, pegylated anthracycline doxorubicin. So I guess what I'm saying here is biology, understanding the biology of complex disease systems is vital for rational therapies. Cells, bodies, disease pathways are extraordinarily complex, and we should not be scratching the surface because we may be playing with fire, and patients who should be benefiting are not benefiting, or worse, coming to harm when it could be avoided. And the message I really want to leave you with from this part of my talk, and I'm nearly done, is this type of fundamental biology exploring the properties of disease systems at resolutions from atomic to molecular complexes to cellular to in vivo is vital to develop full understanding of extraordinarily complex systems. And without this understanding, it is not possible to really develop the next generation of therapies for human diseases like cancer. So our realization that this was the case, and this is not rocket science, I think a lot of people are coming to the same conclusion, has led me over the last 5 years in our Cambridge lab to really begin to focus on what I call congruent barriers. Now my argument is quite simple. I think the same problems, the same practical problems are being encountered both in understanding fundamental diseased systems, as well as in developing rational therapies. What are those problems?
Well, two that are vital, I think, is in order to understand these systems, you have to be able to perturb them and look at the effects of perturbations across multiple dimensions.
And for that, I don't think, despite all the recent advances in genetics, that genetic manipulation is quick enough to perturb cellular systems with biochemical reactions occurring within seconds or minutes. Therefore, there is a crying need for us to be developing approaches to target with small molecule probes, not drugs, probes, the ability to selectively modulate the some 200,000 or so functionally significant interactions that are estimated to exist within the human proteome. And also to be developing new methods to be able to monitor the effects of modulation. And in our Cambridge lab, we have been attempting just this, setting up in collaboration with chemists, structural biologists, physicists, and engineers at the University of Cambridge, platforms to be able to develop chemical probes against a wide range of different structural classes of target. Again, in what we call the systems microscopy initiative, new methods in dynamic microscopy to be able to study biochemistry of reactions inside living cells encoded in the photophysics of fluorophores. And I believe that this type of understanding will enable us to develop new therapeutic approaches, because to develop new drugs, you have to answer exactly the same questions. Where do you drug a particular pathway? What is the best target? You have to know about what the targets are, the rates of the reactions, and how they proceed. How do you drug that target? You have to know how to make selective chemical probes. Who do you drug? Which patients will benefit? You have to know what the effect is of the modulation of the system. And I think overcoming these barriers is really a major goal. And from Cambridge, I have been lucky enough to be able to start, just as of last August, an extension of our effort in the new Center for Chemical Biology and Therapeutics here in India at INSTEM NCBS. It's an interinstitutional collaboration between INSTEM and NCBS, funded in the first instance by the Department of Biotechnology. And we hope that we can move some of this work and extend it here. But why should we care?
And I'm going to end with just this point, which I hope for those of you who have a long and successful future ahead of you as scientists will be in a position, I hope, to appreciate and work in rapidly evolving new practices in biological sciences. These types of problems, as I've said, they are not going to be solved by biologists alone, chemists alone, physicists alone, engineers alone.
These need targeted efforts which combine people, skills, in flat, organized, multidisciplinary teams. This is not going to be easy. And the reason is, as we've already encountered in Cambridge, how do you find common languages between so many different skill sets? How do you harness the innovation that can come from each discipline in a common management structure?
Most importantly, academia at the moment is geared to me.
I did this, I published this paper, I'm the first author, I'm the last author. That doesn't work in these team-oriented exercises. How do we overcome these barriers to allow academics who are talented and ambitious people to sublimate their individual aspirations towards, if you will, a greater goal? These are all challenges. We certainly have no magic solutions, but I think these are the problems we as a community are going to have to solve if in the future we tackle these big problems in science, in biological and biomedical science. I think there's a lot to learn from industry, and I've worked over the last decade with many biotech and big pharma companies because I think they have some very good and efficient ways of doing things. And some of them are summarized here. I won't go into them further. But I'm going to end really, for lack of time, in quoting a maxim from Andrew Grove, who is very famous, of course, in the computing and microprocessor world, where he argued that business success contains the seed of its own destruction by breeding complacency, which in turn breeds failure. And he concluded that disruptive change actually occurs when the paranoid survive. I'm going to be somewhat heretical and suggest that rather than the paranoid surviving in our biomedical environment, what we need is for the collegiate to survive and flourish. And I hope that you will all be part of this revolution, which I honestly believe is coming in the biomedical sciences. So I'll end there and simply say that the work that I'm talking about was done in the Medical Research Council Cancer Unit. It wouldn't have been possible without the very generous support of the Medical Research Council throughout the term of this project, as well as the Wellcome Trust. And this is us. Thanks a lot for listening.